This next portion of our class, we're going to focus on proteins. I thought we would begin with a brief conversation about some of the structures of proteins and their function within the cell. And then we can move on to talking about amino acids themselves, which are the building blocks of our proteins. Talk about the R groups and how they confer the chemical properties of particular amino acids. And then we'll talk about how to form a polypeptide chain, bringing those building blocks, those amino acids, together. All right, so let's jump in. Our learning objectives for the next few minutes is the following three things. At the end of our discussion, you should be able to list several key functions of different proteins. You should be able to explain the structure of amino acids and differentiate between their various chemical properties based upon their R groups. And then lastly, you should be able to show how amino acids are joined together to form a polypeptide chain. So we'll come back to these learning objectives at the end. I want to make sure that these key concepts are solid. So when we go to define what exactly is a protein, quite simply, it's just a polymer of amino acid monomers. We know we have 20 different amino acids, and how those amino acids are linked together, the order in which they are linked together, is going to determine how that particular polypeptide is going to fold. So all those different amino acids, when they're brought together, they're going to start to be attracted to other amino acids and repel other different amino acids, and that's going to give a particular polypeptide chain a 3D structure. And that structure is going to determine how that particular polypeptide chain, how it behaves and how it functions. When we talk about the basic atoms that comprise a protein, it includes carbon, hydrogen, oxygen, nitrogen, and then for some of our amino acids, sulfur is going to be important as well. Before we move on to me actually explaining several functions of proteins, I want you to get out a piece of paper and try to list at least 3 functions of proteins in the cell. Go ahead and pause the video and take a moment to do that. Okay, so hopefully you were able to list several key functions of proteins in the cell, and I'm going to go through several of them now. One of the best places to start when discussing proteins and their function within the cell is to highlight the role of enzymes. Enzymes are the reaction catalyst in the cell. They are responsible for all of our metabolic reactions. They are what convert from one product into a different reactant in the cell. If you think about biochemical pathways, here we have several biochemical pathways within the cell. In the coming weeks, we're going to talk about cell respiration, in which we go from the conversion of glucose. Here we've got the Krebs cycle, and we're going to convert that into ATP, into energy. Each of these blue proteins here is a different enzyme, and these enzymes are highly specific and can catalyze only one particular type of reaction. So we have many enzymes that are able to catalyze each of these specific reactions. A second key function of proteins is the role that they play in the structure of the cell itself. So proteins can make up the cytoskeleton. So think about breaking down this word. Cyto means cell, and skeleton, you're familiar with that word. So this is literally the skeleton of the cell itself. So here we've got the nucleus, and this would be the cell membrane. And now you can see all of these different fibers. We're going to talk about the cytoskeleton in the coming weeks. We've got our microtubules, our actin filaments, and these are absolutely essential to providing structural integrity to the cell. And things will move along the cell through these fibers. Things stay organized because of these fibers. They actually can facilitate movement of the cell itself. We not only have proteins that help to maintain the inner structure of the cell, but we also have things called extracellular matrix proteins. So these are found outside of the cell. Our extracellular matrix is a network of material that is secreted from animal cells and it forms a complex network outside of the cell itself. This is really important for providing strength and structure and support to the cell as well. The third major function of proteins is in the movement of the body itself, or the cell and cell components. So when we talk about movement of the body, you can think about your muscles. Those are made up of myosin, which help to provide contractile forces of your muscle cells. The cell itself can move because of different proteins when it moves through a particular environment. Motor proteins within the cell itself can allow for the transport of different vesicles, nutrients, etc. from within the cell. So proteins are absolutely essential for movement of everything from within the cell to the cell itself, as well as the body. Fourth role of proteins is in regulating cell function. So within this, we have everything from protein, hormones, different cell surface receptors that can be responding to the extracellular environment and initiating a signaling cascade within the cell. We'll talk more about that later on in the semester. We also have transporter proteins that can allow the transport of things from within the cell to the outside of the cell, or vice versa, such as moving nutrients into the cell or moving waste products out of the cell. We also can have intracellular signaling molecules. So within the cell itself, we can have these protein-protein interactions that take place where signals are getting transmitted from within the cell, from one part of the cell to another part of the cell. Antibodies are also proteinaceous in terms of their structure. So we've talked a lot about antibody production as it pertains to COVID, and all the serological tests are looking at antibody production. That is all protein. Blood clotting factors are also incredibly important, and they are proteins as well. They're essential component of the blood to make sure that if you happen to have a wound, that the blood itself will clot, and then the healing process can begin. Another function of protein is keratin as well as collagen fibers. Those help to maintain the elasticity of the skin, and also helps to form a protective barrier so that bacteria do not enter through the skin, that we have this nice layer there to protect us. So now that we've gone through the key functions of proteins, we're going to discuss a bit about the structure of proteins. And that discussion will begin with highlighting the structure of the basic building block, the amino acid. So what I'd like for you to do now is go back to that piece of paper and force yourself to draw an amino acid. The name gives you a clue as to what it includes. It's going to include an amino group and some sort of acidic group. So go ahead and take a minute and see if you can draw that out. All right, so hopefully you drew a structure that looks something like this. So you should be able to recognize an amino acid. If you are shown a bunch of macromolecules, that should be recognizable to you. You should be able to, that central carbon, also referred to as the alpha carbon, is going to have 4 covalent bonds, which you'll see here. So we're going to have a hydrogen that interacts with that central carbon, the amino group, the side chain, which is going to be what confers the specificity for each of the different amino acids, and then we've got the carboxyl group, or the carboxylic acid. So under physiological, pH of 7.2 under physiological conditions, that hydrogen that was part of this hydroxyl group is lost, whereas the amino group, this NH2 group, has accepted a hydrogen at physiological pH. So again, you should be able to draw this from memory and replicate this. and be able to identify the particular amino acid in the context of a polypeptide chain. One last thing I want to point out is we refer to the amino group with the central carbon and the carboxylic acid group. We refer to this as our backbone of the polypeptide chain. We will oftentimes describe the impact of the side chain on the folding of the protein, and there we're really only talking about the R group. If we say the backbone, again, we are implying the amino group, that central carbon, and the carboxyl group. All right, so now let's go ahead and look at some of the specific amino acids. These are our non-polar amino acids. So again, you should think back to that electronegativity table. In order for something to be polar, you're going to have a high difference in electronegativity between the atoms that you're looking at. What you'll notice here is that we're predominantly looking at carbons and hydrogens. So you see that here. Here we've got carbons and hydrogens for the most part. And so these fall within our non-polar amino acids. Now, I don't expect you to memorize these, but I do expect that you would look at something like a CH3 group as the R group and recognize that that would be non-polar. Okay. So in contrast, we have our polar amino acids. We have those that are polar and uncharged and those that are polar and charged. So let's look first of all at the uncharged polar amino acids. So again, a high difference in electronegativity. If we're looking at carbon versus oxygen, there's going to be a big difference in that electronegativity, so we would classify this as polar. Here again, you're going to see that hydroxyl group. We see an oxygen here again. So that's all good indicators that what we're looking at is a polar amino acid. So at this point in time, we don't expect that you will be able to draw this particular amino acid, this tyrosine, from memory. You probably will have to do that once you take biochem. But we do want you to be able to look at a structure like this to recognize that that hydroxyl group is going to make this particular amino acid behave in a polar fashion. And again, you should be able to look at this, identify your central carbon, identify your amino group, the carboxyl group, and then the R group, as well as that hydrogen. All right, so that's polar and uncharged. And lastly, we've got the polar and charged R groups. So the charged amino acids are easy to identify because you can see the charge. Here we've got the negatively charged amino acids. These would be acidic. They gave up their H. This had been a hydroxyl group. They've given up that H, the hydrogen, under physiological conditions. And so that increase in H plus concentration in the particular environment that this amino acid found itself in, now we've increased the H plus concentration, which means the pH has gone down. Contrastingly, we have a series of basic amino acids, and here you can see that the amino acid has been able to obtain a hydrogen, and so now it's going to have that positive charge. So here we've got an NH plus or NH3. Here we have that NH2 plus. And so, in effect, that in order for this particular amino acid to accept that proton, you see a decrease in the H plus concentration of the environment that this particular amino acid found itself in, which would cause an increase in the pH. So this would be basic. This is acidic. These are polar uncharged and are polar charged. So again, if you should be able to go ahead and look at these structures and be able to differentiate whether it's polar or nonpolar, and if it's polar, whether it's uncharged or charged. All right, so please make sure that you can kind of quiz yourself and look at these structures and that you're able to differentiate between them. All right, so we want you to be able to understand the basic structure, meaning that central carbon, the amino group, the carboxyl group, and as well as the R group, and then how that's going to dictate how that particular amino acid, what are some of its chemical properties. Be able to classify the differences between the different amino acids and understand their general properties of each class. Again, don't spend time memorizing this or memorizing the abbreviations. We are not expecting that for this particular class, although you might have to do that in the future. All right, so I want you to take a look at these 3 different amino acids, aspartic acid, histidine, and leucine, and I want you to answer the following question. Which of these 3 amino acids would bury itself deep into the interior of a water-soluble protein during folding? So go ahead and pause the video and take a second to answer this question. So in order to answer a problem like this, it's important to go ahead and analyze each of these amino acids. So the way, again, I would recommend doing that is identifying your central carbon, which is here. Each time, go ahead and take the time, oh goodness, to identify your central carbon. And then we've got our carboxyl group here, we've got the amino group here, and now what we'll want to do is go ahead and look at our side chain, which is here. This is going to immediately, you should recognize that we are looking at a difference in electronegativity between the carbon and the oxygen, so that this aspartic acid would actually be polar, and it's going to be polar and actually charged. So at physiological pH, this hydrogen would be lost, and we'd be increasing the H+ concentration to the surrounding environment, and this would be charged. The side chain here you should recognize as being charged as well. Instead of having a negative charge, this one actually shows you it's got a positive charge. So this one also would be polar and charged. This amino acid, on the other hand, we are looking at the R group here. We've got a bunch of carbons and hydrogens. You know by now that looking at the electronegativity table, there's not a huge difference in electronegativity between carbon and hydrogen. So this would actually be nonpolar. I'm going to go ahead and write NP here. So we've got 2 that are polar and 1 that's nonpolar. And so let's go back and look at the problem. Which of these amino acids would bury itself deep in the interior of a water-soluble protein? So the key idea here is we've got, if you think about the context of a cell, and if we're going to have a protein that's folded here, and what portion of the amino acids are going to be nested on the inside of this protein versus which amino acids are going to be found on the outside of this protein. And we know that like dissolves like. So if we're in an aqueous environment, which is what we find in the cytosol, then the amino acids that are going to be on the outside of that protein are going to be those that are polar. So we'd actually find histidine, aspartic acid on the outside of this protein, whereas leucine, which is nonpolar, is going to flee. It's hydrophobic, it's nonpolar, it's going to flee away from that water, and so it's going to be buried on the interior of the protein. Now that's a really hard problem, and so if you struggled a little bit with that, that's okay, but you should start to try to think your way through these types of problems. Now that we've highlighted the structure of an individual amino acid, I want to end talking about what happens when we bring 2 amino acids together. All right, so here what you can see, we've got glycine, which is 1 amino acid. Here we've got our central carbon, our carboxylic acid group, our amino group with the hydrogen, and the R group for this particular amino acid is simply a hydrogen. Alanine, on the other hand, we've got our central carbon, our hydrogen. We've got a methyl group, that's going to be our R group. Here's our carboxyl group, the amino group. And so what happens when we bring these 2 together to form a polypeptide chain? What we're going to form is water. We're going to lose 2 hydrogens here off of the amino group and an oxygen off of the carboxyl group, and we're going to form what is known as a peptide bond. So this is going to be a dehydration synthesis reaction. So here, now that you can see we're forming a peptide bond, you see that taking place between the carboxyl group and the amino group. And so now that we're going to be building this long chain, it's going to be much more challenging to actually identify the peptide bond. So again, the best way to do that is to find your central carbon orient yourself by finding the amino group, the carboxyl group, and then your hydrogen, and then you can identify the R group. So once we really start to bring together several amino acids, we're going to get much more complicated structures. So one thing I want to point out to you is that eventually, once you have these huge long chains, what we're going to find is that we're going to have what we call the amino terminal end. So this is called the N terminus or the amino terminus. And on the other end is going to be our carboxyl group. So we call this the C terminus. When we're actually synthesizing a new polypeptide chain, we are adding on each additional amino acid onto the C terminal end. Again, this is going to be our polypeptide backbone going along from the amino group, the central carbon, the carboxyl group, etc. And then we've got those side chains, which are really what determines which particular amino acid we are looking at. All right, so I thought we would end by doing one more question. So I want you to go ahead, take a look at this, pause the video, and see if you can answer the following question. Which of these letters shown in red corresponds to the peptide bond? So go ahead and pause this and write down on a piece of paper which you think it is, A, B, C, or D. All right, so again, the key here is to find your central carbon and go from there. So if we're looking at this structure here, you can see we've got our central carbon. Off of the central carbon should be an amino group, a carboxylic acid group. We have a hydrogen, and then we've got our R group. So this is technically our first amino acid, and so the peptide bond would be taking place here at C. Here we've got our next central carbon, alpha carbon, the amino group, the carboxyl group, the R group, and so the peptide bond, again, between these two amino acids is occurring at C. So in summary, I hope that after watching this video, you feel like you can highlight some of the general functions of different proteins. I hope that you feel comfortable identifying the structure of an amino acid and can really differentiate between the chemical properties of the amino acids based upon their R groups. And then lastly, when we bring those amino acids together and form a polypeptide chain, I'm hoping that you can identify where the peptide bond actually takes place.